Dear student, in this video we are going to discuss about the transport across cell membrane. Now why actually this transport across cell membrane is important? What is its implication in our normal physiological process? From this cartoon you can see that this person is suffering from some kind of diarrhea, and he was unable, so much loss of water from his body that he was unable to move out of the washroom. Okay, so this person is going to have dehydration. Now what will be the remedy of this dehydration? The first line of treatment that we give is the oral rehydration therapy. That is, we give the ORS solution and the person drinks it. So this is the remedy. Now how this ORS is going to help this dehydration? So when the person is taking this oral rehydration fluid, it will enter into its gut, and from the gut, from the intestinal cells, these will be transported into the extracellular space where actually there is lack of water. So there is transport of this fluid across the membrane, across the cell membrane. So this is the transport of sodium chloride, glucose, whatever that is present in the ORS solution. Okay, so that is the practical implication. This transport is very much important in our everyday physiological processes. Transport of food, transport of oxygen, everything, it is related to the transport across cell membrane. From our previous knowledge, we know that cell membrane is a biological membrane. It is semi-permeable in nature. It separates the cell interior from the exterior. Now, this model of cell membrane that we see on the right hand side, it is known as the fluid mosaic model. It was proposed by Singer and Nicholson, and they explained the components of cell membrane and their functions. Now, according to this fluid mosaic model, the fluid component is a lipid and the mosaic component is the membrane proteins. So, the cell membrane, it has got a lipid bilayers and the membrane proteins. Now, what are the different types of lipids present over here? There are three types of lipids present here in the cell membrane. One is the lipid with phosphate groups attached with it. So, they are known as the phospholipids. Phospholipids are the most abundant lipids in the cell membrane. In addition to that, they have the lipid molecules where lipid with carbohydrate group attached. For example, they are known as the glycolipid. We can see this. This is the glycolipid. And we have also the cholesterol. So, phospholipid, glycolipid and the cholesterol. These are the lipid formations. Now, these phospholipids, they are the main building block or the fundamental building block of cell membrane. These phospholipids, they are amphipathic molecule and amphipathic means they contain both the hydrophilic and the hydrophobic end. Okay, so their fatty acid, so we can see these phospholipids, they have the polar head and the tails. These tails are the fatty acid tails which are very poorly soluble in water. That's why they are buried into the interior of the membrane. Whereas the hydrophilic ends, which are polar heads, which are exposed to both the sides, that is the extracellular fluid and the intracellular fluid. So the polar heads, that is the hydrophilic end, it is exposed towards water, whereas the fatty acid tails, which are hydrophobic, they are buried into the interior of the membrane. So this is about the phospholipids. Now regarding the glycolipids, mostly these glycolipids are present on the outer surface of the cell membrane. So these lipid molecules, they are distributed unequally. From it we can say that glycolipids are mostly distributed on the outer surface, whereas some lipids present on the inner surface of the membrane. For example, phosphatidyl inositol, phosphatidyl serines, they are present on the inner side. Regarding the phospholipids, there are mostly 6 types of phospholipids present in the cell membrane. phosphatidylcholine, phosphatidylserine, phosphatidylinositol. Then we have the cephalin or phosphatidylethanolamine, sphingomyelin, and the cardiolipin. So the three lipid components are the phospholipid, glycolipid, and cholesterol. In addition to that, cell membrane has the proteins, including the integral proteins and the peripheral proteins. These integral proteins are very important in case of transport across the cell membrane. Now come to the classification of transport across cell membrane. There are mostly three types of transport. One is the vesicular transport, which is further subdivided into exocytosis and endocytosis, passive transport, and the active transport. Active transport is again divided into primary active transport and the secondary active transport. So we will start with the passive transport across the cell membrane. Passive transport means in this transport process, no energy is required, no ATP is hydrolyzed. There are two types of passive transport. One is diffusion, another is osmosis. Diffusion is further subdivided into two types. One is the simple diffusion, another is the facilitated diffusion. So we will start with the first type of passive transport, that is diffusion. What is diffusion? Diffusion means movement of molecules from area of high concentration to area of low concentration. In this picture we can see that at one side there is a fluid with solution with high concentration. So on the other side there is a low concentration solution. So there is a movement of fluid from high concentration to low concentration. So as it is happening along the concentration gradient, so no energy is required for it. Okay, so how long this movement will continue? Until and unless both the side will come to an equilibrium. Okay, so and the time required for the equilibrium by diffusion is proportional to the square of the diffusion distance. It is proportional to the square of the diffusion distance. We know that there are two types of diffusions. One is a simple diffusion, another is the facilitated diffusions. What are the similarities between these two type of diffusion? In both the type of diffusion, it is a passive diffusion, so it does not require any energy. So the movement of fluid will happen from high concentration to low concentration. In both the cases, in case of simple diffusion and in case of facilitated diffusion. So now how actually they differ? In case of simple diffusion, it occurs through the phospholipid bilayer of the cell membrane, and it mainly happens through the non-polar for the small and non-polar particles. This simple diffusion happens. Whereas facilitated diffusion, it requires a carrier protein, a transmembrane carrier protein. This blue thing, this transmembrane carrier protein is required for it. And this type of transport, it is mainly for the large particles or the polar particles. Okay, so the similarity is no energy and both from the along the concentration gradient. The difference is in case of simple diffusion, it is no transport or carrier protein is required, but in case of facilitated diffusion, the carrier protein is required. And in case of facilitated diffusion, the process of diffusion, it is fast. It is faster than the simple diffusion. And also we will see in the next slide, the facilitated diffusion, they follow the saturation kinetics, but in case of simple diffusion, no such saturation happens. We can explain this diffusion, two types of diffusion, simple and facilitated diffusion, by these two simple examples, real life example. Suppose some person are coming down through a slope of a hill. Okay, this on the left hand side, these two persons are coming down. On the right hand side, another person is coming down through the slope. What is the difference? They are coming down on the left hand side, they are coming down by walking, whereas on the right hand side, this person is coming down with the help of a skate. With the help by doing skating, the person is coming down. So who will come faster? Obviously the person with the skate will come down faster. So this is the example of facilitated diffusion, and these skates are the actually the carrier proteins. They are helping the faster movement. Okay. But what will happen after certain time, when suppose instead of 1% there is 100% and only 10 skates are available. So we will see that although the number of person it is high, so the as the available transport proteins or carrier proteins are not adequate, so after certain time there will be a kind of saturation. So in case of the facilitated diffusion, the process of diffusion is faster, but after certain period it will attain a saturation. That is the saturation kinetics. We will see it by the graphical representation in the next slide. Here in this slide we can say that in this graph the concentration is plotted along the x-axis, and along the y-axis the rate of diffusion is plotted. So this green straight line is representing the simple diffusion. We can see as we increase the concentration there will be simultaneous increase in the rate of diffusion. Okay, but in case of facilitated diffusion, after certain period we will see that there will be a phase of saturation. When all the carrier proteins or the transported proteins they are saturated, they are not available. So in that time, at that time, a further increase in concentration on the one side of membrane will not enhance the rate of diffusion. So it will come to a saturated phase. So this saturation kinetics is the speciality of the facilitated diffusion. So what are the similarities between facilitated and simple diffusion? Both are passive diffusion. Along the concentration gradient, no energy is required. In case of facilitated diffusion, we require the carrier protein. Mostly the larger particles and the polar particles, they are transported by facilitated diffusion. It is faster diffusion, but it follows the saturation kinetics. Now the magnitude of the diffusing tendency that can be expressed by this fixed law of diffusion. The magnitude of diffusing tendency from one region to another separated by a membrane. This membrane may be a cell membrane, it may be a blood glass barrier, is directly proportional to the cross-sectional area across which actually this diffusion will take place. And the concentration gradient, that is the chemical gradient, okay, which is the difference of concentration of the diffusing substance divided by the thickness of the boundary. Now this is a fixed law of diffusion where J is equal to minus dA and into the delta C by delta X, where J is the rate of diffusion, D is the diffusion coefficient, A is the area, and delta C by delta X is the concentration gradient. And this D or diffusion coefficient, it will depend upon the particles solubility divided by square root of the molecular weight. Now why the minus sign is given? This minus sign actually indicates the direction of diffusion. When considering movement of molecules from higher to a lower concentration, this delta C by delta X, it will become negative. So multiplying minus dA with this minus delta C by delta C X, it will give a positive value. And the permeabilities of the boundaries across which the diffusion occurs, it may vary in different part of the body, but diffusion is still a major force that affects the distribution of water and solutes. One point I want to highlight here, the facilitated diffusion can be via the ion channels or via the transporters. So there are certain differences between the ion channels and the transporters. Ion channels are the pore forming membrane proteins that allow the movement of ions through the cell membrane, whereas transporters are the transmembrane proteins that transport ions across the cell membrane against the concentration gradient. And the ion channels, they transport ion through the concentration or electrochemical gradient, whereas it actually transported, they do it against the concentration gradient. Now a passive transport, so when we are talking about the facilitated diffusion, so we are mostly talking about the ion channel, movement of ions through the ion channels. When we will discuss the resting memory potential, before that I will make another video on the certain ion channels which are responsible, which are very important to understand the concept of resting memory potential and action potential. Now a passive transport mechanism here happens, whereas in through the transporter mostly the active transport mechanism happens. Now these ion channels, they may be the voltage gated ion channels, ligand gated ion channels, and for example it may be the ion channels like sodium ion channels, it may be the acetylcholine ligand gated sodium ion channels, like this. Whereas the transporters, it may be the primary transporters, symporters or antiporters are the three types. Okay, so there are basic differences between these ion channels and the transporters. Now come to the second type of passive diffusion, that is osmosis. Osmosis is actually the movement of solvent across a permeable membrane. So it is nothing but a type of diffusion of the solvent molecules. What do you mean by that? For example, this is a concentrated solution that has the water particle and some sucrose molecule, sucrose or glucose molecule. Okay, so we know that this is a sucrose solution. So we know the sucrose concentration is higher. So sucrose is a solute here. So normally what happens in case of diffusion, the sucrose will move from higher concentration to lower concentration. Okay. But if we separate this sucrose solution via a semi-permeable membrane, okay, if we separate it via a semi-permeable membrane, at one side we place the water, other side we place the sucrose solution, and the membrane is not permeable to sucrose. So what will happen? But it is permeable to water. Then water is the solvent molecule. So water from higher concentration, it will move to the lower concentration. So movement of solvent particle from higher concentration to lower concentration is known as the osmosis. So osmosis is also a passive passive type of transport where no energy is utilized, because here the solvent is moving from high concentration to low concentration. So the concentration gradient of the solvent is important. But along with it, osmosis is proportional to the reflection coefficient of the membrane also. Okay, so what is reflection coefficient? It is the ability of the membrane to reflect the solute. It is known as the reflection coefficient. That we told that membrane is not permeable to solute. It is not permeable. But whether it is totally impermeable or some permeability is there, so that will be defined by the reflection coefficient. And this reflection coefficient, it actually varies from 0 to 1. Okay, so if the membrane reflection coefficient, it is 0. Reflection coefficient 0 means the membrane is permeable to, it is totally permeable to the solute. No reflection is happening. Okay, so it is totally permeable to the solute. That means there will be no osmosis here. And if the reflection coefficient means 1, that is complete reflection, all the solute particles will be reflected back. That means in that case there will be the osmosis will actually happen. So the reflection coefficient is a number between 0 and 1 that describes the ease with which a solute permeates a membrane. If the reflection coefficient is 1, the solute is impermeable. Therefore, it is retained in the original solution. It creates an osmotic pressure and it causes the flow of solvent or water flow. Now if the reflection coefficient is 0, that means solute is completely permeable. Therefore it will not exert any osmotic effect and it will not cause water flow. So the osmosis, we can say, it is proportional to the concentration gradient of the solvent as well as with the reflection coefficient of the membrane. Now the tendency of the particle, solvent particle, now the tendency of for the movement of certain solvent particle to a region of greater solute concentration. So to the region of greater solute concentration. So in this direction the movement is happening. The solvent is greater here. So it will move to this direction. Now this can be prevented. The movement of this solvent can be prevented by applying pressure. By applying pressure to the more concentrated solution. So if we apply pressure here over here, and these hydrostatic pressure necessary to prevent the movement of solvent is known as the osmotic pressure of the solution. Okay, so the hydrostatic pressure necessary to prevent the osmosis, the movement of solvent from high to low concentration, is known as the osmotic pressure. So when we are studying osmosis, we need to know about two things. One is the osmotic pressure, and osmotic pressure it depends upon the concentration of the osmotically active particle. So we want to know how we will express the concentration of osmotically active particle. So one thing is the osmotic pressure, another is the concentration. These two things we need to know. Now osmotic pressure, it is like the boiling point elevation or freezing point depression. It is a colligative property. Okay, so it depends on the number of particles rather than the type of particles in the solution. That is, it is a fundamental colligative property of the solution. In an ideal solution, osmotic pressure P is equal to, it is nRT divided by V, where P is the osmotic pressure and N is the number of particles, R is the gas constant, T is the absolute temperature, and V is the volume. Now if the T is held constant, it is clear that the osmotic pressure, it is proportional to the N by V, that is the concentration. We can also call it the N by V, we can write it C. So P is equal to C or T, the concentration of the particles. What type of particles? That is osmotically active particles per unit volume of solution. So now we must know how can we express the concentration of osmotically active particles per unit volume of solution. Now in chemistry, if we want to express the concentration of any solution, we will say it in terms of molarity, that is number of moles per liter. We know that the mole of 1 mole is equal to 6.023 into 10 to the power 23 number of particles. That is the Avogadro's principle. So now weight of 1 mole in grams, that is the atomic or molecular weight of that element in grams. For example, in case of sodium chloride, we will see that we know that the 1 mole of NaCl will be equal to their molecular weight will be 23 gram plus 35.5 gram. It is around 58.5 gram. Okay, and we know that 1 millimole is 1 by 1000 mole, 1000 of a mole. So we can convert it into this molar concentration into millimolar concentration. So 1 mole of NaCl, it is 58.5 gram. So 1 millimole will be equal to 58.5 milligram. Okay, so per liter can by this way we can express the concentration. But if this sodium in our body, inside our body, this sodium chloride will be in a solution. We have the blood plasma, where this sodium chloride will be ionized into two osmotically active particles. One is sodium another is the chloride. So the pressure exerted by this osmotically active particle, the concentration of this osmotically active particle, it is better to express it in the terms not moles but the osmoles. Now what are the difference between these moles and osmoles? So there are difference if the particle is osmotically active, that means the sodium chloride. So when it will go to the solution, it will there will be two osmotically active particle, one is sodium, another is the chloride. Okay, so their concentration of osmotically active particle, it is doubled. But in case of glucose, you will see that glucose is not ionized, so the number of moles will be equal to number of osmoles here. Okay, so it is not osmotically active, so it will not ionize. So 1 mole of glucose will give rise to the same osmoles of osmotically active particle. Hence, to express the concentration of a solution in terms of number of particles, the unit called the osmole. It is used in place of grams. Now, 1 osmole is 1 gram molecular weight of osmotically active solute. Thus, 180 gram of glucose, which is 1 gram molecular weight of glucose, is equal to 1 osmole of glucose, because glucose will not dissociate into the ions. Conversely, if a solute dissociates into two ions, 1 gram molecular weight of solute will become 2 osmoles, because the number of osmotically active particles is now twice as great as in the case for the non-dissociated solute. Therefore, when fully dissociated, 1 gram molecular weight of sodium chloride, we know that we calculated, it was 58.5 grams, that is 1 mole NaCl per liter. It is 58.5 gram per liter, that is equal to 2 osmoles. Similarly, we know that that 1 mole per liter, millimole, if we express it in 1 millimole, then it will become 58.5 milligram. So 58.5 milligram is equivalent, will be the 2 milliosmol of sodium. Okay, so we actually express them in terms of osmoles per liter or osmoles per kilogram. So here we will come across with the two terms. One is the osmolality, another is the osmolarity. So number of osmoles per liter of solution, osmoles or milliosmoles per liter of solution, is known as osmolarity, whereas number of osmoles or milliosmoles per kilogram of water or solvent is known as the osmolality. As 1 kg of plasma is equivalent to 1 liter in our of plasma in our body, we usually use this term osmolarity, that is number of osmoles or milliosmoles per liter of solutions. Now, how can we measure the osmolar concentration of plasma, or we can say the osmolarity of the plasma? Now, osmolarity can be measured by this freezing point depression. What is this? Now, the presence of solute in a solution, it lowers the freezing point of a solution relative to that of the pure solvent. For example, pure water, we know that it freezes at 0 degree centigrade. If one dissolves 10 grams of sodium chloride in 100 ml of water, the freezing point, it will go down to minus 5.9 degree centigrade. So in case of pure water, the freezing point is 0, but we make, if it is a solution containing some solute, that there will be depression of the freezing point. Now, what is the freezing point depression of plasma? If we know it, then we can calculate the osmolarity of the plasma. It is seen that if we dissolve 1 mole of any substance in any solution, so the freezing point depression, it becomes 1.86 degrees centigrade. So 1 mole per liter or 1 osmole per liter, we can say that is equal to 1 mole means 1 osmol, that means 1000 milliosmol per liter. Okay, so we can say 1000 milliosmol per liter of a concentration of a solution, it depresses the freezing point by 1.86 degree. So 1 milliosmol per liter will depress the freezing point 1.86 divided by 1000, that is 0.00186 degrees Celsius. So we can say that the number of milliosmoles per liter in a solution equals to the freezing point depression divided by 0.00186 degrees Celsius. So that will give us the osmolarity. How it is giving osmolarity, we'll see. First, you remember that if we dissolve 1 milliosmol of solute in 1 liter of a solution, the depression of freezing point of that solution will be 0.00186 degrees Celsius. Now experimentally we have seen that blood plasma, it freezes at the freezing point depression, it is freezes at minus 0.54 degrees Celsius. So the freezing point depression is minus 0.54 in case of plasma. So we know that if the concentration is 1 milliosmol per liter, then freezing point depression is 0.00186 degree. So what point we can say in this way, 0.00186 degree depression of freezing point means 1 milliosmol per liter concentration. So 0.54 degree centigrade of depression of freezing point means 0.54 divided by 0.00186. Obviously, so here we can say that the osmolarity of the plasma will be 290.3 milliosmol per liter, or we can say it 290 milliosmol per liter. It actually varies within a range 290 to 300 milliosmol per liter. So from this freezing point depression, we have generated the value of plasma osmolarity, that is 290 milliosmol per liter. Now by measurement of serum osmolarity via this freezing point depression method, it is an experimental method, but clinically we usually use this formula, that is osmolarity is equal to 2 sodium, that is sodium is expressed in milli equivalent per liter, plus glucose concentration of our body, milligram per deciliter, divided by 2.8, plus blood urea nitrogen level divided by 18. So by this formula, clinically we have to measure the sodium concentration, glucose concentration and blood urea nitrogen concentration, and if we have these values available, so we can calculate the osmolality of the plasma, which is very much needed to know the hydration status of a patient. Now we know the osmolarity of plasma. From this we can calculate the value of osmotic pressure of the plasma. Okay, by this Van 't Hoff's formula. So from this formula we get that osmotic pressure of 1 milliosmol per liter is equal to 19.3 millimeter of mercury, and we know the osmolality of plasma, it is 300 milliosmol per liter. So 300 into 19.3, it is 5790 millimeter of mercury. It is a calculated value of osmotic pressure, but actually if we measure it experimentally, it comes around a bit less, that is 5500 millimeter of mercury. Why this difference happens? Because many of the ions in the body fluid, such as sodium ion, chloride ion, they are highly attracted to one another, so they cannot move entirely unrestrained in the fluid and create their full osmotic pressure potential. Okay, so that is the reason there is a gap between the calculated value of osmotic pressure and the measured value of osmotic pressure. So there are so many osmotically active particles available, solutions available which are needed. For example, the hypertonic saline that is usually given when there is very low sodium in the patient. So these osmotically active particles, they are hyperosmolar particles, they are transfused. And if this osmolarity it goes beyond this range of 300 milliosmol per liter, there will be some conditions like hyperosmolar conditions, and it may cause severe fatal effect on the patient. For example, in case of uncontrolled diabetes mellitus, there may be hyperosmolar hyperglycemic state that can lead to diabetic coma. So osmolarity is a very important parameter. It has to be maintained within this narrow range of 290 to 300 milliosmol per liter. There are so many osmoreceptors and a feedback mechanism present in our body that controls the osmolarity of our plasma. Some weak acids and bases, they are soluble in cell membrane in undissociated form. But when they become dissociated, it becomes difficult for them to cross the membrane in this charged or dissociated form. So what happens, how they cross the cell membrane? The molecules of the undissociated substances, they diffuse from one side to the other side of membrane, and on the other side they then dissociate. Okay, so this type of diffusion is known as the non-ionic diffusion, and the process by which ammonia is secreted into the urine and then it is charged to NH4 plus, it is an example of non-ionic diffusion. We have completed the first part of the transport across cell membrane, that is the passive transport. Now come to the active transport. Active transport processes, they utilize the energy by hydrolysis of ATP. Okay, so there are mostly two types of active transport. One is the primary active transport, another is the secondary active transport. In the primary active transport, the energy is derived directly from the breakdown of ATP. What are the examples of primary active transport? Most important example is a pump, that is sodium potassium ATPase pump. Okay, other examples, so many these transporters are mostly called the pump because they are pumping against the concentration or the electrical gradient. Other examples, it may be H plus K plus ATPase pump in the gastric mucosa and in the renal tubules. It may be the calcium ATPase pumps that pumps out the calcium out of cells. H plus ATPases acidify many intracellular organelles, namely the lysosomes. So there are certain examples of primary active transport. Now in this video we will specially discuss about the most important pump of our body, that is sodium potassium ATPase pump. Now sodium potassium ATPase pump, it uses the energy to move 3 sodium out of the cell. We know from our previous video, that is the body fluid distribution and the ionic concentration, we know that extracellularly the most higher concentrated ion is the sodium ion. So extracellular sodium concentration is high and intracellular potassium concentration is high. Okay, so here the sodium is moving from already the sodium is high at the extracellular space, so instead of this fact it is moving against the concentration gradient. So what should be the normal tendency of the sodium? To move from outside extracellular to intracellular, but as the concentration is higher at the outside of the cell. But here with the help of this pump, the sodium is moving, 3 sodium ions are moving out of the cell, so against the concentration gradient. And we will also see that intracellularly it is negative and extracellularly it is positive. So when the normal tendency will be this intracellular negativity will try to retain this positive sodium ion inside. Okay, but against the electrical gradient also the sodium is moving outside. Okay, so the sodium is moving against its concentration and the electrical gradient. Now with the help of this pump. And another function this pump does, that it moves potassium particles, it moves 2 potassium ions from outside to inside. We know that at the intracellular part the potassium is already high, so concentration of potassium is high, it is inside. So normal tendency of potassium will be it should move from inside to outside, but against this concentration gradient 2 potassium ions are coming inside. Okay, so what is the function of sodium potassium ATPase pump? It utilizes energy to move 3 sodium ions from inside to outside of the cell and move 2 potassium ions from outside to inside the cell. But in case of potassium, the movement is against the concentration gradient but along the electrical gradient, because inside it is negative, it will obviously attract the positively charged ion. Okay, so we can say that the coupling ratio of this sodium potassium ATPase pump, it is 3:2, that is 3 sodium outside and 2 potassium is going inside, and it is an electrogenic pump, which means it moves 3 positive charges out of the cell for each 2 that it moves in. Okay, that means the more number of positive ions are going outside and only 2 positive ions are coming inside. So we can call it as an electrogenic pump. Now if we want to see the structure of the sodium potassium ATPase pump, you will see that it is actually heterodimer. It has 2 subunits. Both of the subunits are different. It has 2 subunits. We can see one is the alpha subunit, another is the beta subunit. Alpha subunit has the intracellular and extracellular binding sites. The intracellular portion of alpha subunit, it has a sodium binding site, that is 1. It has a phosphorylation site, that is 4. And it is an ATP binding site, that is 3. And the extracellular side of this alpha subunit, it has potassium binding site, that is 2, and the Waben binding sites, that is 3. Whereas the beta subunit is a glycoprotein which has 3 extracellular glycosylation sites. Now how this actually functions, we will see it in the next slide. How this sodium potassium ATPase pump acts: when the sodium ion or sodium, it binds to the intracellular binding site of the alpha subunit, ATP also binds to the phosphorylation site, and ATP is converted to ADP, with the phosphate being transferred to aspartate 376 and the phosphorylation at the phosphorylation site. So we have seen that intracellularly we had the one is the sodium binding site, one is the ATP binding site, another was the phosphorylation site. So when sodium binds, so ATP also binds and it is converted to ADP, and the phosphate will be transferred to the aspartate 376 at the phosphorylation site. So that will cause a configurational change. Okay, so this causes a change in the configuration of the protein, and it will cause the extruding sodium ions into the extracellular fluid. So it is move, the some configurational change will happen, and so that the channel is now open on the extracellular side and the sodium ions will go out. Okay, now then what will happen? Then in the extracellular space, what happens? The potassium will bind extracellularly at its binding site, and there will be dephosphorylation of the alpha subunit, which returns to its previous conformation and releasing the potassium inside the cell. Okay, so first attachment of sodium, then hydrolysis of ATP, then the phosphorylation, then configurational changes or conformational changes, and it will extrude 3 sodium ions outside. And then the potassium ion will attach, there will be dephosphorylation of alpha subunit, it will return to its previous conformation and it will release the 2 potassium inside the cytoplasm. So in this slide, by animation, we can see, so first the sodium ions are attaching, then the ATP will be hydrolyzed, the phosphate will be transferred, and there will be phosphorylation and the configurational changes. The sodium ions will move outside, then the potassium ion will come and they will attach, bind to their site. There will be dephosphorylation, phosphate is going out. Then again it will come to the original conformation and potassium is released inside. Now some important information regarding the regulation of sodium potassium pump. The active transport of sodium potassium pump is one of the major energy using processes in the body. On an average, it accounts for about 24% of the energy utilized by cells, and in neurons it accounts for around 70%. Now there are certain hormones which enhances the activity of the sodium potassium ATPase pump. One hormone is most importantly, that is a thyroid hormone. Another is the aldosterone. Another is insulin. They will increase the activity of the sodium potassium ATPase pump. On the other hand, cardiac glycosides like digitalis or digoxin, it will inhibit the activity of the pump, and OABN also, it will also inhibit the activity of this pump. Dopamine in the kidney, it inhibits the sodium potassium ATPase pump by phosphorylating it and causing the natriuresis. So these are the regulators of the sodium potassium ATPase pump inside our body. The second part of active transport is type of active transport is the secondary active transport. We have already gone through the primary active transport. The example was sodium potassium ATPase pump. Now come to the second type, that is the secondary active transport. In primary active transport, I told you that it utilizes the energy by hydrolysis of ATP directly. Okay, so it directly uses the ATP, breakdown of ATP, and takes the energy for the movement of ions against the concentration or electrical gradient. Here actually in secondary active transport, it is an active transport. It requires the energy, but not directly at the surface of cell membrane where this transport process is happening. The best example you will see in case of renal tubular cells or the intestinal cells. So here actually what happens, these luminal cells, the cells in the intestinal lumen or the renal tubular lumen, they have got two surfaces. We can see here one surface that faces the lumen, that is a luminar surface. Another surface that is opposite to the lumen, which actually faces the capillaries or blood. This is the basal surface. So all these luminal epithelial cells, they have got two surfaces. One is the apical surface or the luminal surface. Another is the basolateral surface or the basal surface. So what happens? When we take some food or anything, actually the transport process, it occurs through the lumen across this luminal surface of the cells. This cell membrane will take part into the transport. Okay, the energy that will be utilized, it is not at this luminal surface from where actually the absorption is taking part. So where it is now utilized? It is seen that in the basolateral surface, what happens, the energy is utilized via the sodium potassium ATPase pump. Okay, so what happens, suppose I have taken the ORS which contains the sodium and glucose. The best example is SGLT or sodium dependent glucose transporters. Okay, so what happens, when the glucose and sodium is coming into the gut, the entry of glucose, it is coupled by the sodium. Okay, so how it happens at the basolateral surface, actually the sodium potassium ATPase pump is acting, it is utilizing energy, it is utilizing ATP. Okay, and as a result of the sodium potassium ATPase pump, we know that its function is to expel 3 sodium outside the cell. So intracellularly the sodium concentration will go down. Okay, and along the concentration gradient by passive way, this sodium will enter into the cell. Okay, the concentration gradient is created by the sodium potassium ATPase pump, which is acting as the basolateral part. But from the surface where actually this transport is happening, here no energy is utilized. Here there is passive transport along the concentration gradient of sodium, and when the sodium will enter, it will take the glucose with it. That's why it is known as the sodium dependent glucose transport, or SGLT. Okay, so this secondary active transport, it is an active transport. Here the energy is required, but it is not at the surface where actually the transport is happening. Okay, that is the example. They can be co-transporters where direction of ion movement is same. Now it can be the antipode or exchangers where direction of ion movement is opposite. So it is an active transport, but it is secondary. The best example you will see in gut and the renal tubule where there is sodium dependent glucose transport happens. Now the last type of transport is the vesicular transport. We have gone through the passive transport, this diffusion and osmosis. We have learned about the osmotic pressure and osmolality, and then we have gone through the active transport and the primary and secondary active transport. So this is the last portion that is remaining, is the vesicular transport. Vesicular transport can be of two types. One is the exocytosis, that is vesicles are going outside the cell, and endocytosis, vesicles are going inside the cell. And when it is moving inside, it can be the movement of a solid particle, that is known as phagocytosis, movement of a liquid particle, that is cell drinking or penocytosis. Another important type of endocytosis, that is the receptor mediated endocytosis. First is the exocytosis. It is a calcium dependent process where the cellular contents are moved out of the cell in the form of vesicles. Now the proteins involved in the exocytosis, actually what happens when the cells they try to move these vesicles they actually fuse at the cell membrane. Okay, and this fusion, it involves certain proteins. What are those proteins? That we'll see in the next slide. So there will be a docking process of these vesicles or the docking or fusing process of these vesicles with this cell membrane. Okay, so some proteins which are present on the vesicular side, they will form a bond with some proteins which are present on the cell membrane side. Okay, and this docking process will be facilitated by the presence of calcium ion. So in the vesicular side, we will see the synaptobrevins are there. Okay, so the synaptobrevins, they are present over here, whereas syntaxin, it is present on the cell membrane side. Okay, so there will be a bond formation between this synaptobrevin and syntaxin, and this bond will be helped by some other types of proteins or the snare proteins, which are mainly useful for exocytosis of hormones like insulins or neurotransmitters like acetylcholine. Okay, so this process, when this bond will be formed, so these vesicles they will ultimately go outside the body, outside the cell. The 2013 Nobel Prize in Physiology or Medicine was awarded for the discoveries of machineries regulating vesicle traffic, and this scientists Rothman and Shakeman and the Seeldorf, they were awarded the Nobel Prize in Physiology or Medicine in the year of 2013 on this topic of vesicular transport. There are two modes of vesicular release by the exocytosis method. One is the regulated pathway, that is also known as non-constitutive pathway. Another is the constitutive pathway. In non-constitutive pathway, proteins from the Golgi apparatus, they initially enter the secretory granules where processing of prohormones to the mature hormone occurs before exocytosis, and it is called the regulated pathway. And constitutive pathway, it involves the prompt transport of proteins to the cell membrane in vesicles with little or no processing or storage. Okay, so when the processing is there, so that is a regulated or non-constitutive pathway. When there is no processing, this is the constitutive pathway. One of the important clinical implication of this V snare and T snare proteins are the Clostridium botulinum toxins. They actually inhibit the release of acetylcholine by blocking these snare proteins. Okay, botulinum toxins has been weaponized by some terrorist organizations for the bioterrorism. So that will cause, if the acetylcholine release is blocked, that will cause the flaccid paralysis. Okay, at the nerve terminals there will be no acetylcholine and there will be flaccid type of paralysis. There are several isoforms of botulinum toxin, 7 types: A, B, C, D, E, F and G. Among them, A and E, they block the SNAP25. A and E, they actually block this SNAP25. Whereas toxin BDFG, they cleave the synaptobrevin. BDFG, they cleave the synaptobrevin, and toxin C, it cleaves the syntaxin. Okay, so SNAP25 will be blocked by toxin A and E, and BDFG will block the synaptobrevin, and C will cleave the syntaxin. Okay, so this is the clinical importance of V snare protein and T snare proteins in the exocytosis. After exocytosis, we are coming to endocytosis. Endocytosis is the reverse of exocytosis. So there are various types of endocytosis depending upon the size of the particles. But here in this video, I am discussing about the clathrin mediated or receptor mediated endocytosis. This process, which transport the substances from outside to inside the cell and uses the protein clathrin. Now, clathrin molecules has a shape of triscallions with three legs radiating from the central hub. And clathrin, along with the GTP binding protein dynamis, is involved in the process of this clathrin mediated endocytosis. Now, as the endocytosis progresses, we can see here the clathrin molecules form a geometric array. It forms a geometric array, and this surrounds the endocytic vesicles. Now at the neck of the vesicles, we can see here at the neck of the vesicle, the GTP binding protein dynamin is involved, either directly or indirectly, in pinching off the vesicle. Once the complete vesicle is formed, the clathrin falls off and the three-legged proteins recycle to form another vesicle. Now the vesicle fuses with the dumps it contains into the early endosomes. Okay, so from the early endosomes, a new vesicles can be barred off and return to the cell membrane. Okay, so this is a type of clathrin mediated endocytosis. It is responsible for the internalization of many receptors and the ligands bound to them, including, for example, nerve growth factor, low density lipoprotein. It also plays a major role in the synaptic function. These are the references, and thank you.